Hello everyone. Today me, Rabi Atif, and my partner Farhanaz Khan will be presenting a paper on the discovery of conserved regions in DNA sequences by the ABC, which is the artificial bee colony algorithm. I would first like to break down the title for you. A conserved region in evolutionary biology is a sequence of either DNA or RNA that remains unchanged in a species throughout the evolution. Also, this conserved region is something that is maintained through a natural selection process. Problem statements and aims. So what we're trying to do here is that we're trying to find a conserved sequence, and we're going to do that by using a swarm intelligence based technique, which is the artificial bee colony algorithm, and we will be investigating the results of two different modifications, mod 1 and mod 2 of this same ABC algorithm. So why is this important? This is important because conserved sequences, they hold a lot of importance in biology due to the fact that they are used in processes like translation and transcription. In translation, what happens is that the genetic code is decoded to produce amino acids, and in transcription, information in the DNA is copied into a new molecule. Moreover, these conserved sequences are very useful in bioinformatics, and they're specifically used to find similarity among different organisms and species. Thirdly, there has been research done on finding conserved sequences, but the traditional approaches that we have, they're time consuming and they do require considerable amount of effort. So in order to find a more improved solution, we have this ABC algorithm. We'll now discuss the basis and the concept behind this algorithm. So what happens is that in the initialization phase, we have a random generation of food sources. After that, our employed bee phase starts. In our employed bee phase, each bee is assigned a food source. That bee goes to its assigned food source and exploits its neighborhood, and it updates the new best if the neighborhood source is better than the originally assigned food source. After this phase, we have the onlooker bee phase. In the onlooker bee phase, the employed bee comes back and shares the information with the onlooker bee. The onlooker bee then uses a probabilistic selection and finds a food source, goes and exploits it, and then repeats the same processes by updating the new best. After this onlooker bee phase, we have the scout bee phase, and in the scout bee phase, the scout bees, they discover new and unvisited food sources, and if a food source is not worth exploiting anymore, then the employed bee that was associated with that food source becomes the scout bee, and then that scout bee goes on a hunt for another food source. And lastly, in the end, the best food source so far found is registered. So now we'll see how this ABC algorithm is adapted to our DNA and conserved regions problem. So in our initial population, Rn is the number of different sequences, and in this particular case we have four different sequences. We have the length of each sequence, and the length of each sequence is 15, as we can see, and the length of the conserved region here, we can see that it's 5. So now we need to make, the only thing that we need to see here is the starting position. So this position vector that we have, it indicates the starting position, the randomly generated starting position of each sequence. So this 6 basically corresponds to the first sequence, and from here we take 5 indexes since our length of the motif is 5. So from here we take C, A, T, A, A, and we put that in our alignment matrix, and we repeat that for all the 4 sequences. After our alignment matrix is made, we then make the profile matrix. In the profile matrix, we have our 4 bases, the 4 DNA bases, which are the A, T, G, C. And in this, what happens is that we take a column-wise frequency of the bases that we have. So in this first column that we can see, we have 3 C's and 1 T. So here in the first column, we have 3 C's and 1 T, and this is how the profile matrix is made. After the profile matrix, we have the consensus sequence, in which we have the best result from the profile matrix. So for example, in the first column, we have 3 with C with the highest frequency, so we will choose C here, and this process is repeated for all the bases. We will now move on to Farhanaz Khan to discuss the rest of the process.
So now we move on to the employed bee phase for our first modification. So, firstly, the food sources are randomly assigned to employed bees, and then they start to explore the neighborhood by using this equation. Here, xi is the current food source, that is the current position vector, and phi is a random number between 1 and -1. Xk is a randomly generated position vector, and j is also a random number between 0 and length of the motif. So basically what we're trying to do with this equation is that we're changing one element of our current position vector to produce a new food source. As we can see here, this is our current position vector, and our j came out to be 4. So we're putting the jth element of our current position vector in this equation with the jth element of our randomly generated position vector, and we get 8 value. So we change the jth element's value to 8. So this becomes our new food source. For our second modification, we're basically not using that equation and only swapping the jth element of our current position vector and with our randomly generated position vector. So for our fitness calculation, the employed bees follow this function. So if the objective fitness is greater than 0, then it adds 1. But if it's less than 0, then we scale it to a positive number, basically avoiding negative values. So our objective function, fitness function in this case is similarity function, which uses the maximum frequency of each column in the profile matrix. For example, in this column, the maximum frequency is 3, which is of C basis. So max P I equals to 0 would be 3 for our zeroth column. So now in the onlooker B phase, our actual food source is selected by this probability function, which is similar to FPS that we have studied before. So food sources with higher fitness values have a higher chance of being selected. And the scout B phase is carried out by using a parameter called limit, which is the number of cycles after which the food source should be abandoned, making the corresponding employed B a scout B. So for the experiment, the paper chose three different data sets with different values of N and L. The number of food sources was 100 and the number of iterations was 3000. These fixed values were tested against different motive lengths and different values of the limit parameter. So this table is for modification 1, and we can see that as the length increases, the mean and best fitness values are decreasing. And we can also see that for modification 1, limit equals to infinity produces better results than limit equals to 50. Limit equals to infinity means that we are skipping the scout B phase and we are not abandoning any food source. But this is opposite for modification 2 because limit equals 50 gives better results than limit equals to infinity. Mod 2 was also found to be more exploitative than Mod 1, and we can see from these graphs where this bold line is Mod 2 and this dotted line is Mod 1, and this was tested across different lengths, such as 10, 12, 13, and 18. And we can see that across all these lengths, mod 2 converged faster than mod 1, even though mod 1 also generated similar results eventually. The paper also briefly discussed a parallel implementation of both of these modifications. So in the parallel version, the population, which is the food sources, is divided into subpopulations, um, and on all of the ABC operations are performed in those subpopulations in parallel. The paper found out that the parallel version produces better results in less time, as we can see from this particular graph, where the bold line is parallel mod 1 and the dotted where the bold line is mod 1 and the dotted line is parallel mod 1. And this is for length equals 9, and we can see that parallel mod 1 produces better results. The paper also compared the algorithm with multiple existing evolutionary approaches, such as a G-based algorithm, a D-based algorithm, and also some ABC-based techniques, and it found out that most of the time, the proposed algorithm with mod 1, mod 2, and their parallel versions showed better accuracy than other algorithms. These bold values express best results, and we can see most of the times it is the proposed algorithm. So in conclusion, mod 2 was more exploitative than mod 1, even though mod 1 also eventually produced similar results. This means that mod 1 is more explorative, and the paper also preferred mod 1 in that regard. We also saw that parallel implementation of these modifications produced better results in less time, and this entire proposed algorithm with modifications and parallel versions was generally found to be better in accuracy than other existing algorithms. Thank you. If you have any questions, you can ask.